Thank you very much, and thank you for the invitation today. And actually, it's been a great 2 days. Just hearing about something else than flu has been fantastic. But I will take you back to flu right now, because it is still my favorite subject. Now, who has had the flu this season? Yeah, just a few of us. Is it all the middle-aged people? No? Yeah. But yeah, so it's been pretty bad this season. If we compare to the graph here, and you can see the 2009 pandemic, although the numbers hadn't been as high as we would have expected, it was an unusual emergence because it happened during the off-season, what we would consider the off-season. But we can see in 2017, 2018, so our season, how high flu was. And when we talk about flu, we're always, because of the vaccine we have and how we have to develop a new vaccine every year, we're always thinking about prediction. Can we predict the next flu strain? And so this is a typical tree that you would see where you have the phylogeny of the different types of strains and how genetically the virus mutates such that every season you have new genetic clades that emerge. Now here I'm showing you the pandemic H1N1, so the 2009, what we call the pandemic H1N1, and antigenically it's very similar, but genetically it has mutated. And so here in the X's, you can see the vaccine strain. So this year there was from last year, a change in the composition of the vaccine, where a new genetic or antigenic variant had been selected. But again, looking here, we're looking at consensus, and that's what is circulating over the population. And for many years now, we've been starting to look at within-host diversity. I've been harping on that because what you see emerging in the population on a global scale has to start from individuals, and that's where your mutations happen. Now, yesterday we heard about short-sighted evolution, and I thought that was very interesting because here we are dealing with an acute virus, and flu does a lot of diversity. It's a segmented genome, so it does reassortment. It has that high error rate, so you have a lot of accumulation of point mutations. And all this will happen within your host and then will be transmitted. And we've heard about transmission yesterday. Now, because it's an acute infection and it occurs usually typically over 7 to 11 days, you have a very limited time for that evolution to happen before transmission occurs. And then there's that bottleneck reducing the diversity. But what if flu, you looked at flu in a chronic type infection? And there are cases where if you have a severely impaired immune system, you could have a long-lasting infection. And in those hosts, the virus probably evolves in a different way. And here's an example of a study. Actually, this was published quite a while back, but the last author, Guy Boivin, had asked us to do the sequencing, the deep sequencing of samples that had been collected over 2 years. The main question being, can an individual be infected with the same virus over 2 years? So it's like a chronic infection, which we don't see with flu. Obviously, it's an acute infection. And so when we did that, we actually did see that here you have a cluster of all those samples, and they only clustered with themselves. And this was everything from previous seasons and seasons later, and those really clustered with themselves. And this is a representation of all the samples that would cluster into clades based on their date of collection. And so you can be infected for a long time with flu, and when you start looking at what is going on in that infected host, what was interested is that we know flu does reassortment, but we can never catch it within individuals because it's an acute infection. But here we could see reassortment that had happened within that individual. And these are just these trees that show with the different clustering and the colors whether the samples cluster from the different segments. It has 8 segments. And here you can see that you have possibly reassortment based on how these different samples clustered. And so this is the first instance of the evidence of reassortment within a host. But we can't catch that during an acute infection. It would be very difficult to do. Now, flu does, like I said, a lot of mistakes. So what we tend to look at are these single nucleotide variants. And when we talk about diversity within the host, that's usually where we focus. And I'll talk to you about also defective viral genomes a little later. And you'll see where it all comes together. And so in the nucleotide variants, what we look at, again, is that deep sequencing, what seems to be emerging, ideally over time. And in fact, when you look at a study, here it's a study we did with Brazil samples. These are different seasons. Your x-axis are different individuals that were sampled. And what I represent here are different positions on one of the surface protein, the hemagglutinin. And the nucleotides here are their relative frequency. And what you can see there is that some individuals have mixes of 2 nucleotides, and that's simply that there's probably another little strain that's in the co-infection. But what is striking is that these individuals from 2011 are carrying already minor variants that emerge later on in the season and become dominant. So could we use deep sequencing for surveillance to predict what will emerge in the future? Possibly, but here, you sort of have to know already when you're surveilling what is fit enough for the future. So we may not be able to predict, and it looks great in hindsight, but would be difficult going forward. What was interesting when we looked at these samples is that, in fact, they did emerge as the dominant clades in the world for H1N1. So these different positions that I'm showing here, they're single positions, but they appear to all go with one particular strain emerging. So I'm talking about the within-host diversity, but of course there's transmission that has to occur for these strains to emerge in the population and circulate throughout the world. And it's capturing that diversity and determining what is being transmitted that is an aspect that we've talked about in these past few days. And here, the diversity in your recipient will often be determined by the diversity in your donor, but also by the bottleneck. And the bottleneck has been a little bit difficult to assess because it depends on your host. Now, the diversity from your host is dependent on certain aspect of the host that I want to go into. We usually talk about diversity and the bottleneck in a very generic way, where we consider sort of every host the same, but in fact we're not all the same. And I'll show you some examples where your immune competence can determine the diversity of what you're carrying. When I showed you that chronic infection with flu, it was actually driven, the reassortment was driven with antivirals, a lot of antivirals in that child that was infected for 2 years. And that will determine also the diversity and what is going on. And also we've been exploring, and I won't go into that, but even the respiratory microbiome can influence the diversity of the population of viruses you carry. And so, as an example here, it's the comorbidity, if you're obese or not obese, can also have an effect on how long you're shedding the virus. So how long you're infected, how much you're shedding, will have an impact on that bottleneck and on what is being transmitted to the recipient. And we started a study with Stacy Schultz-Cherry, some of you know her here, she's at St. Jude, and she developed a ferret model of the obese, and she calls them her pudgy ferrets. And so what she noticed is that actually transmission was much faster and easier in her fat ferrets. And she had this one particular subtype that does not transmit by aerosol in the lean ferrets. It transmits really well by contact. So the ferrets tend to touch each other. They play like kids, and so they have a lot of secretions and sneeze on each other. But some of these strains or subtypes do not really transmit well by aerosol. But it did in the fat ferrets. So is it that the fat ferrets are more susceptible, or are these shedding more virus? And so when we looked at the different diversity, and it was a very limited set, but we did find a lot more minor variance in the fat model of the ferrets than in the lean ferrets, and there was more transmission that occurred of these. We couldn't calculate the bottleneck, the transmission, because we had a very limited set. But you can see how the host can impact that diversity and can impact the transmission. Another aspect, we're not all equal. We're all different ages. The older you are, the more you've been exposed at some point in the past to flu. And you are carrying that history of your immune response to flu. And the younger you are, the less you've been exposed to it. Now, it's difficult to study this in the human population because often people don't remember if they've had the flu. They think they've had the flu. You can do some antibody testing, but there's cross-reactivity, so it's never really clear what you've been exposed to in the past. But we can do it in a more controlled way with ferrets, and this is what we did with Ted Ross at University of Georgia, where he basically took ferrets and infected different groups of ferrets with different decades of H1N1 viruses. The idea being that maybe being exposed to a certain strain will determine how you respond to a subsequent infection. And maybe you would push the evolution of that virus differently depending on what you've been exposed to in the past. So these ferrets here, called pre-exposed, were actually exposed to any of these four strains I list up here. And then we're put in contact with naive ferrets. But first, actually, they were pre-exposed for 12 weeks. We left them alone, so they cleared the infection, and then they were infected with the California 01, that 09, so that's the pandemic H1N1. Then they were put in contact with naive ferrets, and then we had pairs that were purely naive. And so we had 46 ferrets across 23 pairs, so we were able to look at what was going on in these pre-exposed ferrets versus the naive ferrets and see with what impact that immune exposure, so that exposure to the virus could have had on the evolution of the H1N1. And so this work is being done by Kate Johnson, and some of you, she's right here, some of you may have met her at her poster yesterday, so I will be presenting part of her work here. And her big question was, does flu diversity differ between a naive or a pre-exposed group? And that's where we'll start talking about these defective viruses I mentioned. And so here this just shows the diversity of the mutations that you find in the infection at different time points, but here all together in naive ferrets or pre-exposed ferrets, and these would be the directly infected ferrets, versus their contacts, and so who they're transmitting it to. And there was no difference really at the diversity level depending on what you've been pre-exposed to in the past. And here we had a bottleneck of about 4 to 6, which seems to be what others have found for ferrets in general. But when we look at the HA, you see the diversity going down from the pre-exposed donor to the recipient, but we see the same thing in the naive. And we see this bottleneck here, and that's normal because you are basically restricting what is being transmitted. But here you're seeing more of a bottleneck in the pre-exposed because probably it's clearing some of these viruses. So this all looks the way you would think it should look. So here I'm showing you this because I'll be using these colors. So I have these naive, what we call the naive ferret pairs, where they've never been pre-exposed, just infected. And then you have the pre-exposed donor, but it's still a naive recipient. One of the questions here is that if the host determines the diversity of the virus, would these naive look similar, or would the naive look more than like its donor? And so what we see is that, in fact, when you cluster based on the diversity and what happens in the distance between each of the samples, so how closely they look to each other based on their diversity, all the pre-exposed, be it the donor or the recipient, this one being naive, will cluster together so that your diversity is often determined by the donor. So all that pressure probably was done by the donor on the virus before it's transmitted. So you have that bottleneck that already has determined what will take root in the recipient. And that's what we saw here. Now, if we look at the diversity at specific positions, actually here, so it's hard to see because the graphs are not showing up, but we have all our ferrets that correspond to our pre-exposed pairs and all the naive ferrets, and they're sampled at multiple time points. And this shows that only one group of ferrets right here had a mutation that emerged, none of the other ones. And what was interesting is that that was on an antigenic site of the hemagglutinin. And in fact, it was only the group that had been pre-exposed to that strain where two of the pairs developed that mutation. So could it be that in the population, depending on your age, depending on what you were exposed to as your first flu, could determine what emerges later on? So could an older population push the virus evolution in a different way than a younger population? So these are aspects that we haven't explored yet, but it's possible. And so here, with that type of mutation, that antigenic variant, or that sort of the genetic antigen at an antigenic site, did emerge in the population, the human population, when we look at what the consensus of flu that's been circulating since 2012. And so here I focus mostly on nucleotide variants, but during an infection, be it when you get the flu or my ferrets, we actually also have a lot of defective viruses that are generated. And it's thought that in an infection, about 90% of what's transmitted is defective. So very few of those viruses would be able to replicate on their own and would be, whole, basically, have all 8 segments that are there. And so one type of defective virus that it makes, it's where the segments have a big deletion in the middle, but the ends are conserved. And the ends are necessary for replication and also for packaging into the virions, so that all these defective particles are actually still packaged. All these defective genomes are packaged and can be transmitted. And so we've looked at this here. You will see in flu that most of the defective segments are in the polymerase. And so the polymerase, actually here I represent my segments as these little sausages, but in fact they're intertwined here. The RNA has the nucleoprotein around it and then the polymerase complex. So that as long as that polymerase complex is there, even if this is defective in a virus, it will use that protein for replication. So that you see in a flu infection, if you have a low MOI, low multiplicity of infection, you will have usually just a wild type competent virus that will be replicated. And if you have a defective virus that enters a cell on its own, it will not be replicated because there's no functional polymerase. But if you do a high multiplicity, where you would have a lot of the competent virus and maybe some of these defectives, then you would have replication. And in fact, the defective viruses get preferentially replicated because the segments are shorter. The shorter the segments, the faster the polymerase replicates. So in our experiments, we actually can show that these DVGs, or these defective virus genomes, can also be interfering. So that's when we call them DIs. And here I'll just call them DIs. Here's an example where you can put a PB2 DI, so that would be a segment that's got that gap in the middle, and it will hinder the replication of the wild type or competent virus, because it will overtake the polymerase machinery, and basically a lot of the DIs are being replicated rather than the competent virus. So that's just a standard thing that will happen with DIs. So let's go back to our ferrets. We can actually find a lot of these DIs, and what we do is that we map the reads, and we map them to find the start and the end of the gaps. And here, this is just a representation of different types of DIs, and they're usually in this area where they're about 500 to 1,000, they're 300 to 800 nucleotides long, but here you can see that it's usually at the position 150 to 250 on one end, and the whole segment is about 2.3 kb. And so these DIs have a much shorter segment that is preferentially replicated. And so looking at these DIs, we actually see them accumulating over time, which we would expect if they're preferentially replicated. And we see this happening in our naive ferrets, and there's no pressure to deplete those DIs, but they go down in our pre-exposed ferrets. So they are being selected against because of that pre-exposure before. And we see also a lot of DIs in our contacts, and one of the questions is, are these DIs being transmitted? Here, the stock, they're really, shoved into the ferrets, so of course they would have DIs in the donors, but determining whether they're transmitted is a more complicated thing in this case. But we do see them accumulating very quickly, and when we look at the species, they're very similar. So are these happening de novo simply because it's an error that the virus does systematically, or are they transmitted is not always clear. But what we do see is that some of these DIs, and here they're represented as a relative frequency, and these are all the pre-exposed pairs, and we see some of these DIs that actually have an open reading frame, and some are enriched in the pre-exposed, and some are enriched in the naive pairs. So there are species of DIs that occur depending on the host. And we don't know exactly what they're doing. Presumably, they're probably interfering. And I'll show you some evidence that we do see interference with these DIs. And what Kate has found also is that if we take the frequency of unique DIs, so types of DIs based on these gap positions I was telling you about, we see peaks in the naive where you have more DIs in the naive, and they're shorter in size than in your pre-immunized where you have fewer and they're larger in size. And remember, I told you the shorter you are, the more you get replicated. And if we look over time, we see an accumulation of DIs, and the size is becoming restricted. So there's a decrease in the genome segment length over time of these DIs. And we see this systematically across our ferrets, and here they're just all pooled together, but over time, this is what we see occurring. Now, DIs, we've seen a few years ago, could be transmitted based on evidence where we saw shared DIs, two types of DIs from two individuals that were living in the same dorm in UC San Diego campus during an outbreak. And so there is some evidence that they could be transmitted, but we have to do this more systematically to know what's happening. And if we look at our ferrets, it's very difficult when we look at recipients that have DIs and we just look at what is shared. The size of the circles indicate what is shared or how many species they have, and the size of the line shows how many of these would be shared. Now, what we do see is that the naive look more similar, have more in common than they have with the pre-exposed. So there could be transmission of the DIs that has occurred, but because there were so many DIs in our stock initially, it probably really is being passed on during the infection. But so here I'm showing you DIs in natural infections or in our ferrets, but what is going on really at the single cell level? What do we see? Because again, although that diversity happens within hosts, it also happens within individual cells specifically. And so this is another PhD student who has done some single-cell genomics, and she actually has shown here we can see the clustering of the cell. You capture the cell mRNA and the viral mRNA at the same time. And this shows you just clustering based on what the cell mRNA is, the expression of individual cells, and those are these, each point is an individual cell. And what we do see is that there is different groups here that are expressing a lot of messenger RNA from the virus, and a cluster here that is not. And we'll look at that cluster specifically. But here, if we look at just the host response to infection, it would be a typical host response where we have some indication that there's stress happening in these heavily infected cells or cells that are producing a lot of virus because it's taking over the host machinery. But in these two other clusters where we had much fewer viral reads, we see that the viral genome replication is being sort of put down a bit, and we have interferons, the interferon pathway that is quite high in these two groups. But more interestingly is that where we had in the cluster, we had low viral reads, we actually had a lot of DIs. So systematically, when we have low virus, we have a lot of DIs. And when we look at these clusters specifically, we see again a big innate immune response that in these specific cells. The question is, are these cells responding to the infection and clear the infection and only the DIs are left? Or is it the DIs that are stimulating that response and helping clear the infection? Now, DIs are thought to be sometimes good, sometimes bad. It's not clear whether the virus, it's part of the evolution of the virus to have a lot of DIs to maybe dampen the infection so that it's not as severe, and so if your host is not as sick, you can transmit better. And so these are aspects we'd like to better understand. But we looked specifically at the DIs within these individual cells, and we mapped those reads to see what types of DIs were present in these cells. And we found two that were present at very high abundance, and that's typical of DIs where you will find during an infection some DIs are present at a very high level, and then you have a lot of species present at a low level. And we found these two, and they have sort of a loose border on either end, but we decided to make these viruses and see whether they would be interfering. And we do see these are, in yellow, are ones from the border that we call the DI222. And in purple would be the other DI. And this would be your wild-type virus. And when we put DIs with the wild-type virus, it basically prevents replication of the virus. So these are truly interfering viruses that we see within these single cells. And what was interesting is that in our cluster where we had a lot of DIs, the majority of the DIs were actually that DI that was the most interfering. So it's probably the DIs that are really keeping the virus infection down and probably stimulating the innate immune response of the virus. So you can see how complicated it becomes when you start taking into account in these infections not just the minor variants or the single nucleotide variants, but then we're introducing defective viruses that may have a true function during an infection. And so even when we talk about transmission, the DIs may also be playing a role. And maybe a lot of the minor variants that we identify are actually on defective viruses. So that's something else we have to look at when we start generating these haplotypes. So in summary, we do see that the diversity in a flu and an infection will differ depending on your immune background or on any comorbidity or any other aspect that could have an impact on inflammation in some way. And we do find unique variants depending on what your immune history is, and that could be true in the human population too. We just haven't really looked at that specifically. And you can see how the DIs, or these defective viruses, have a different dynamic and is an aspect that we have to take into account when we look at within-host diversity. And DIs exist in other viruses too. I know there are groups working on Zika virus DIs. You see them in all kinds of infections. So as we explore more of the within-host diversity, it would be worth looking at these defective viruses too, because they seem to be playing some kind of role. And so one small aspect I wanted to finish on was sequencing flu is relatively easy. When I hear about these other viruses, it can be far more complicated. It's easy because A, it's segmented, and the segments are sort of the ideal size if you want to do a multi-segment PCR, if you want to amplify the segments for sequencing. And flu conveniently has these conserved ends, and they're conserved across subtypes. And you can have, these little degenerate primers that will capture all subtypes and most of the diversity, or so we hope. And so here you can do just this multi-segment RT-PCR, generate each of your applicons, and then do deep sequencing so that you don't have to worry about what would be the host contamination or any other thing. So you can get quite deep representation of what you're sequencing. But when you're looking at these single segment genomes, these larger genomes, often people have been focusing on specific genes because it's harder to sequence these whole viruses. And so we've heard on, I think it was yesterday, some metagenomics approaches to basically go into the virome and capture in an unbiased way what is there. Well, so here I want to introduce you to Yanqing if you want to contact him. He's at Penn State University, and Yanqing is a biomedical engineer, and he's currently in the lab of a physicist, a materials physicist. And these guys have developed a device, and this is one of the first prototypes, and he's in my lab here in one of the hoods, and we're helping him test multiple viruses on this device, where the idea is that they basically generate these, here I'll show you a little video, they generate these nanorods. They're carbon nanorods, and depending on the distance between the rods, they can capture different sizes of viruses. And he's right now developing one that is a gradient, so that from a sample you could capture different sizes of viruses. And so he's tested this with different types of samples, but for example, he had taken some tissue swab from chickens, and he did the sequencing before enrichment and after enrichment. And this is typical, where you get the host genome, you get ribosomal RNA, and you get a lot of no hits. You just don't know what's in there. And this was after enrichment. He went from a 1% to 90%. So I think this could be a pretty powerful platform. And what's great is that it actually captures the viruses so that you can culture them after. So the viruses are not destroyed when they're captured on the device. And so this is all I had for you. These are, I mentioned Kate, I mentioned Chang, and these are other people in the lab. Bin Zhu was a senior scientist in my lab. He's now at the CDC, and he's team leader for flu vaccine development. And Katya Cole, some of you know, she's basically been working, Kate has been working with her on the DI transmission and evolution, and Anna is a collaborator on the Brazil study that we've been doing. And then I was asked to show this since I'm an editor on microbial genomics, and they couldn't be here at this meeting. But if you want to submit to microbial genomics, here it is. Thank you very much.
Thank you very much for a fascinating talk. There's plenty of time for questions. So
yeah, DI, I mean, are we talking about sort of free RNA within a cell? Or thinking about, because you talked about DI viruses, so is the DI virus, I mean, I'm thinking it could be, you know, it's a virus which is missing part of PB2, so it's got the 8 segments in it, but one of them's shortened. Or it could be a virus that's sort of encapsulated on release from the cell, and within that it's picked up some free RNA from the cell. Sorry, just for my own benefit, could you clarify what are we talking about?
So which one is it? Well, we don't know which one it is. You have the RNA you're extracting. The fact is that you are seeing it in all of the ferrets. So some of these DI types are shared in all of them. They were in our stock. That means they're being replicated. So it's not just free RNA. It has to be replicated, encapsulated. It's not going to be just free floating on its own. And then within the cells, I mean, you can't differentiate what will be encapsulated or not. But presumably, if they have these ends that are conserved, they have everything that's there to be basically put into a virion and then sent out.
Okay, so they're packaged.
Yeah, they could be packaged, but you can't differentiate. Of course, you're going to be sequencing things that are not packaged.
Okay, thanks.
Thank you for your talk. I just have 2 quick questions, perhaps. I'm not sure about one of them. Maybe you could help me. So, so you calculated a bottleneck of DIs.
No, we didn't calculate the bottleneck of DIs. What we did, it was just the bottleneck of using the usual diversity and then determining what that number was. It could be DIs. It could be... Now, we are looking at minor variants. If the DIs don't have any point mutations, we wouldn't capture them as diversity. They would just be, you know, they would look like the, what would be the consensus, basically. So right now, when we calculate bottleneck, we don't know what we're calculating. It could be taking into account defective viruses, like it could be competent viruses.
I see. And my second question, it's sort of a naive question. The dynamics of DIs when they were with the sort of normal viruses, which you mentioned, remind me of some sort of like cooperative behavior, like development of like cheaters versus like cooperators, which like Stuart West just published on sociovirology. Of like, are you thinking about like some sort of modeling, like the dynamics of cheaters versus cooperators, that sort of stuff?
Well, we're not modeling it, but certainly I do think that during transmission, you're talking of more of a cluster of viruses that you have, and then these defective viruses are cheaters, basically. They are, but it could be that the infection transports with it its bag of diversity. Could it be that these DIs are contributing even in reassortment to the diversity of what is happening, while at the same time dampening the infection a bit, so that it's basically an evolutionary process that these DIs exist. We see them actually in all infections. When we start mapping the reads, you can see DIs, basically.
Yeah, thank you very much. I have a very short sort of technical question. To what extent do you think these DIs are preferentially amplified in PCR?
I think they're very much, in PCR, that's a good question. As much as the matrix would be, because they're actually smaller than the matrix, and we do see the matrix not necessarily preferentially amplified. So in PCR, I mean, that's something we have to be careful of, that basically what we see as present at a high abundance, it wouldn't change the diversity that we're seeing, but it could change the abundance that we're seeing. And actually, the first time when we were, actually that paper on transmission in humans, David Wentworth is on that one, and he was convinced that it was, he didn't believe that there were DIs in natural infections, and he thought it was the polymerase that was skipping during PCR. So we actually tested it with 3 different polymerases. We did every possible test, and it was always the same DIs that would pop up. So I know that answers partly your question. Yes, Tom.
Hi, great talk, Elodie. My question is perhaps a dumb one, but did you and Ted also test transmission to pre-immune recipients, or is it too hard to initiate infection in an animal that's already?
No, but that would have been a great idea, but we didn't do it. Yeah, from a naive to a pre-immune. Yeah, that would have been great.
Going back to your single cell work, what are the cell types? Because you have these nice patterns. Is it just that they're specific immune cells?
No, so the cells are, you mean what cell type did we use? It was the A549. But that's what you're asking. But the different groups you're asking. We did this in also in primary pulmonary, human pulmonary epithelial cells. We saw the same thing.
Okay.
I'm just showing the prettier ones.
Okay.
Yeah.
More questions?
All right. Thank you very much.
No. Okay. Thank you very much.